Good morning. Welcome back. I hope you had a good weekend. All right, so we're going to continue with the respiratory system diseases, and we will get started with the cardiovascular system diseases right after. And then we will move on Wednesday and hopefully get to finish our cardiovascular diseases. So we had talked about tuberculosis as it being a chronic illness. And when we looked at this picture, we were looking at basically the development of a type of lesion in the lung, or what will be interpreted as a lesion in the lung. And this is what we refer to as a tubercle. Now, at this point, the patient may be experiencing some mild coughing and some discomfort, but nothing too serious, at least not serious enough that the patient is going to maybe seek medical attention right away. But it can happen that patients start producing actually a lot of sputum, and that may lead them to go to the doctor and get some medical care. Now, that sputum can be cultured. Unfortunately, with Mycobacterium tuberculosis, like with all the mycobacteria, it grows very slowly. So diagnosis via culture can take several weeks. Now, as it is a slow developing disease, hopefully it is caught before what happens with the tubercles can take place, which is the rupture of them. Now, in the picture, they're showing you how bacteria can potentially leave the tubercles and go to the nearby open spaces. And you also have how some of the bacteria may be getting to the bloodstream. And what you need to think about is that then that bacteria can get pretty much anywhere, wherever there's contact with your blood. And it is completely possible for the formation of tubercles in other parts of the body besides the lungs to take place. Now, this is something that's very serious, and this is something that typically we refer to as secondary tuberculosis. We could have these types of lesions in the brain, which can be very damaging. Sometimes you have them in extremities, other organs besides the lungs and the brain. So at that point, recovery is very unlikely. And it has to do with the fact that now you have possible infection points in different parts of the body that are slow growing and that will require treatment. Now, Mycobacterium tuberculosis is one of those bacteria that over time has become resistant to a lot of different antibiotics. Now, in Texas, what we see most of the time is going to be that what is diagnosed in patients is going to be multi-drug resistant, where the typical treatments that we used to use decades ago are not going to work. That's when we used to use the rifampicin and the isoniazid. Now, luckily, we have a lot of different drugs that can potentially be used. And the problem comes from patients that start feeling slightly better, and those patients stop treatment. When they stop treatment and they restart treatment maybe a few months later, what they're doing is basically promoting antibiotic resistance by basically choosing for the bacteria that are naturally resistant to a certain antibiotic that they're being exposed to. Now, it does happen that when the patients do this on and off, then there will be resistance to additional antibiotics. And the worst case scenario is what you see on your screen here, what we're referring to as XDR tuberculosis, where it is very difficult to treat the conditions. And there have been some cases of tuberculosis diagnosed in some Eastern Bloc countries where you have no treatment possible and you just kind of have to wait and see. Basically, it's just a slow, terrible death, really. Now, in the US, we do keep an eye out for tuberculosis. Any diagnosed case gets followed through. In Texas, we're very vigilant about it because we have a lot of tuberculosis. And usually what it's done for those that are going to be working with patients or in a clinical setting is that we have this tuberculin test that is used. The tuberculin test is just looking for antibodies against Mycobacterium tuberculosis. The assumption being that if you have antibodies, either you had tuberculosis or you currently have tuberculosis. So if the tuberculin test is positive, then the follow-up is going to be chest x-ray. Now, for those that have had the Mycobacterium vaccine in other countries, like in the UK and Cuba, for example, you're always going to come up positive in that tuberculin test. So to prove that you do not have active tuberculosis, then it is recommended that an X-ray is done. Are there any questions so far? Lesions. So what are they looking at in the X-ray? They're looking for lesions. Because if they see a lesion, they don't know what it is per se, but in combination with a positive tuberculin test, that is a bad sign. And then they have to follow up if there is a lesion in the X-ray that can be visible with culture. And they're going to try to get sputum out of the patient, or they can actually go and do what we call a lavage of the lungs, which is not pleasant, but it has to happen if there's a lesion in the X-ray. Any other questions? You're welcome. All right, now what we have here is something that we also have a problem with in Texas, and it is commonly known as whooping cough. Now, the disease, the regular name, the medical term for it is pertussis because it is caused by a bacteria called Bordetella pertussis. Now, this bacteria in adults does not cause, really at the most, you may have coughing, you may feel like you're about to get a cold, but it's not terrible in those that are adults. Now, in younger people, especially the young ones like toddlers, newborns, it can actually be deadly. The problem is that if you come in contact with it, it is very likely that you are going to become infected. And the way it happens is that in children, we see in young children that they go from having that cold-like illness to developing a lot of coughing with stringy mucus. And that is because when you have that bacteria in your cilia, the cilia cannot move because the bacteria produces a toxin that stops the movement. So you still have the urge to push something up that's caught in this dry mucus, but you can't really push it upward. And what you have is cases of patients that are constantly coughing, to the point where they turn blue. And in the case of very young children, we see that in many instances, they actually throw up. and there's always that risk of aspiration, especially in the babies. So what we have is that we do prevention in the form of the vaccine. If someone is suspected to having whooping cough, that we used to do culture for this, but it wasn't the best test ever. So what is recommended for diagnostics today is actually a PCR, where we look specifically in that swab, nasopharyngeal swab, for the bacteria, specifically for the genes that make that toxin that is causing all the trouble. Because we're not supposed to have in our bodies any type of bacteria that has those genes. So the presence, the detection of those genes indicates that you have that infection brewing in your respiratory system. Now, the vaccine seems to be pretty successful. There have been some cases where the protection hasn't been outstanding, but there's still some, better than nothing. And for those that have children that are very young, like if you have a newborn, this is one of the reasons, one of the many reasons why it is not recommended that your baby comes in contact with too many people, that your baby doesn't get kissed by too many people, because as adults can have this in the respiratory system, they can potentially pass this along to the baby very easily. Are there any questions? All right, so we're going to move on to inhalational anthrax. Vaccines for adults, no baby. No, the vaccine is available for children. It is totally available for children. In adults, if you are going to be working with young children, or if you know that you want to have kids soon, it is recommended that you get a booster just to lower the chances that you will be the one that transmits this to the baby. There have been several cases. There are several case studies published. You can find them easily online of parents that lost newborns to whooping cough. And it turned out that the parents were the ones that actually had the bacteria in the respiratory system. There's even, I guess it would be one of those Department of Health types of commercials that there was in Oklahoma. There were two couples that were in their commercial a few years back, basically just encouraging people to vaccinate against whooping cough. All right, so we're going to move on to inhalational anthrax or pulmonary anthrax. So previously we discussed how you could have injuries to your skin and have cutaneous anthrax lesions. And these cutaneous anthrax lesions were usually because there were endospores of this bacteria in soil or in fences, in the skin of animals, in the dirt, that you could have. So, well, I mean, it is the same anthrax that causes cutaneous anthrax is going to be the one that gives you inhalational anthrax. It's just that instead of having it on your skin, you have inhaled these endospores of this bacteria into your lungs. So usually what you have at the beginning of the infection is going to be fever and malaise and headache. You feel like you're coming down with the flu maybe, but you have a lot of lung congestion. And when you look at chest x-rays, you'll notice that there's actually an area in the x-ray that shows you the space, the pleural space that basically divides your lungs in half in a way that seems to be just darker than the rest of the lung or even around the lung, as you can see in this picture. And what you have there is just a type of buildup of inflammation fluids that we see typical with this type of condition. Now, technically, it is not transmitted from person to person because you will have to be releasing endospores that get spread out. So what you have inside your lungs, more than likely, if you have the infection, is going to be no longer an endospore, but the vegetative cells. Those that develop inhalational anthrax that we know of, at least in our time, have done so because they were exposed on purpose to powders that contain a whole lot of endospores of these bacteria. Now, how do we diagnose these? Well, there are some very good rapid tests that were developed by the Centers for Disease Control when we had that issue with the shipment of letters and boxes that have the powder in them to different addresses. Normally, for any type of pulmonary issue like this, we may use culture, but the disease moves very quickly, so that's not good enough. So what we try to do is, while we wait for the culture, there's a PCR that is actually a rapid PCR that can give you test results within 6 hours of receiving that sample in the laboratory. And that makes the diagnosis a lot faster. If you diagnose the patient correctly early, you will be able to survive this. The treatment is available. It's actually something that's easy to find in any type of pharmacy. It's ciprofloxacin. It's just that we have to know that that is what we're dealing with. Are there any questions? And I didn't add in here that there is a vaccine. That vaccine is not widely available. It's available to those in the military and to those that work in laboratories where they may be testing these types of samples. And it was, oh gosh, I can't recall how many shots. I can't remember if it was 2 or 3 shots, to be fair. It's been so long since I had it, I can't recall. But I know I had to go in to get my shots more than once. I just had this feeling that it was a series of 3 shots. Now, as far as how many cases there was a question about that. So you're talking about the ones that were related to that local terrorism, because that's what it was, local terrorism attack. Well, there were 8 confirmed cases, but many of them survived. It's just the first few did not because they were not diagnosed properly. Any other questions? So if I can't recall if 2 or 3 died, to be fair, I just can't recall. It's been so long. And there were no side effects of this vaccine at all. I was like, oh, it's like nothing. Yep. Unlike the COVID vaccine, which gives you some very interesting side effects after you get the second shot. This one, for something that's pretty terrible, it was very mild. All right, so we're going to talk about viral lower respiratory tract infections next. We have quite a few, but we're only going to focus on those that we tend to see around in the US. You're probably thinking, we don't see a whole lot of hantavirus. Actually, we see it every summer. maybe not in Texas, but when we see it, it's something that we really need to think about. But we need to also mention that technically, anytime, any single time you have a viral infection that can potentially get to your lungs, you could have pneumonia. So it's not just from your typical influenza infection that you end up with a viral pneumonia. It could be from some of the viral infections that we looked at previously, like, for example, measles, or maybe even if you have an infection with chickenpox as an adult. So it is very difficult to confirm what type of virus is causing the infection if we get the patient once they have developed the viral pneumonia. Sometimes, depending on which virus it is, if we know the history of the patient and we know that they presented with chickenpox or presented with measles, we could maybe very quickly determine that that's what caused the pneumonia. But for the most part, it's going to take some work, and it's going to probably need viral culture to be done to be confirmed a specific virus, unfortunately. Now, we do see that with influenza virus, there is always this tendency, or there's this risk of developing pneumonia from it. Maybe not right away, but if you get very weak from the infection, then it can potentially make it so that you start accumulating fluid in your lungs, and you have inflammation in the lungs, and that would then give you the typical or expected symptoms of pneumonia. Now, at the beginning, influenza starts and almost feels like it is exclusively an upper respiratory infection. But very quickly, it starts migrating down, so you have more of a cough, more of a chest pain, malaise. It could be just that. It could be just the general discomfort, the fever. Or you could accumulate fluid, and then it can turn into a pneumonia. Now, as far as what can we do for somebody that has influenza infection, let's talk about just the mild cases, the ones where you're actually just in bed for several days, you feel miserable. Almost every single person that I've dealt with that have had pneumonia, they always say, oh, I feel like I'm going to die. And I'm like, yeah, it feels terrible. It's not the best feeling at all. You can't work. You can't move around very well, at least for a few days. And every time you cough, you're basically spreading virus to others that are around you. So we recommend that you stay put, you lay down, you take care of your symptoms. If you happen to be very lucky, so that you are diagnosed before the rapid flu test, before you start developing the severe symptoms, then and only then you may be given an antiviral like Tamiflu. Because of the way Tamiflu works, it is useless to give Tamiflu to those that already have the severe symptoms. And we're trying to make it so that we do not have antiviral resistance among the different influenza strains. Now, something that we know happens for those that recover from influenza is that very commonly they get a secondary viral infection or a secondary bacterial infection. And that is because your lungs have gotten a beating, and it is a lot easier for other microbes, even sometimes microbes that were part of your upper respiratory normal flora, to actually cause infection in the lungs because of your weakened state. Now, we do have a vaccine that is provided in the fall, early fall, every year to try to reduce the cases of influenza that we see around the United States and around the world. The way this vaccine is developed is by collecting samples from patients at the end of flu season, toward the end of flu season, so that we can try to predict which are going to be the strains that are going to be going strong at the beginning of the next flu season. There's a lot of math involved in that setup to choose which viral strains of flu are going to be included in the vaccine. Now, the reason why we end up having to do this every year, and it is actually very expensive, trust me, but it would be more expensive if we didn't actually have the vaccine, because there would be a lot of people that could die and also that could not work. It's because this virus, influenza virus, does have a tendency, now you will say it that way because they don't always do it, but we know that it can happen, where you have changes in the proteins in the capsid over time. And when that happens, the antibodies that we made against the previous version of the virus are no longer going to help us. So we actually call that antigenic drift. There's another version of that we call antigenic shift that we'll talk about when we get to a certain bacteria. But what that does to us is that we are basically sitting ducks every year unless we get vaccinated against the newer strains, because the antibodies that we make previously are not going to be able to help us. Now, we do know that some bacteria do what we call antigenic shift, and they can literally just change what they look like when they come in contact with other microbes. And we also have found out that influenza virus can potentially do some antigenic shift as well. And that is that because they can potentially come in contact with other versions of flu virus, and they tend to pick up genes from the other flu viruses that may be around. And that can make it so that you have yet newer, potentially more dangerous versions of influenza. Is that what? There's a question that Daniel had. Is this why we have so many cases a year, or does this have to do with the virus genetic? Well, so this is not going to be a short answer. I had to preface this by saying that. So like I said earlier, we collect samples. Every state lab that participates, that works with the CDC, sends samples at the beginning of flu season, through flu season, and a lot more samples at the end of flu season to the Centers for Disease Control. The World Health Organization also collects samples from around the world. Now, not every country participates, but we at least get a good picture of what's going on. And there's some statistical analyses that are done based on those samples to determine which 6 strains, it varies a number of strains from year to year, can be added to the vaccine or should be added to the vaccine. Now, it has happened. You probably remember the H1N1 outbreak that they became kind of like a pandemic some years ago, where we may have missed the mark as far as there was something lurking at the end of that season that we missed. They usually don't mutate so quickly. It usually takes them about 9 to 12 months. So more than likely that time, we ended up with that flu pandemic type thing. It was, I mean, I call it a pandemic because it was multiple countries that had it. So that does fit the definition of pandemic. And we had a lot of cases in those countries that had it. We actually had just missed something. But it takes them about 9 to 12 months to change significantly so that we can actually basically forget what that virus looked like or not recognize it anymore. Now, the numbers vary year after year. So do the numbers of those that vaccinate themselves, get vaccinated. So it's very tricky to try to establish a correlation. But that time we messed up. We just missed that one. It happens. Now you can still get flu even if you have gotten the flu vaccine. Why do you think that is? Yeah, that's it. That's all it is. It's a strain the vaccine did not have. And none of the strains that were in the vaccine were similar enough to the one that you were so unlucky to be exposed to that it happened. Now, we do have some things that we say to people at the beginning of each flu season. Make sure that you cover your coughs, that you wash your hands, that you don't touch your eyes and your mouth a lot. That does sound very familiar, I know. Because that will reduce the chances of you coming in contact with viral particles for sure. Are there any questions? And notice that in the previous slide, I made a note on purpose telling you that there are no intestinal symptoms. So influenza does not cause diarrhea or cramping. It's just that there's this thing around, it's more prevalent in some states than other, that people refer to as stomach flus, which are not flu at all. It could be stomach virus, but it is not influenza. It's something else. So how often do we have this antigenic shift so that you have human strains picking up maybe genes from the influenza viruses that infect animals? Is that what you were thinking of, Adam? Well, so let's talk about this picture. So you could have, there are influenza viruses that exclusively infect chickens. There are some that exclusively infect pigs. There are some that exclusively infect dogs. There are some that may have some crossover where they may infect or at least multiply in more than one type of animal. Now, those are the ones that may come in contact with some of the human strains. Now, what we need to think about is, and this is a very special scenario here, it does sound like a movie because that's the way we depict it in the movies, but it's true. If you are working with animals in such a way that you are in close contact with them, very close contact, and you could shed influenza virus of the human version to the animal, that's when we may see that the influenza virus that infects the human can come in contact with those that may be already in the animal. Now, that in itself is not a problem, but like I said, it's a very special scenario where where our animal handling has gotten really weird over time, in some countries worse than others. And then other humans may come in contact with those animals, and they themselves get these, I'm going to refer to them as recombined versions of influenza, where they are human influenza that happen to pick up a few genes from those animal influenza viruses. Now, they may be producing new proteins. They may be able to evade the immune response differently. They may have new characteristics that they didn't have before. Now, how often the process of exchange of genes happens? We've tried to account for it, but it's very difficult. But we know that in some countries, it happens a lot more often than others. But what's going to have to happen is that a human picks that strain up from an animal. And then that human takes that strain to other humans. So that has not been measured yet. That has not been calculated yet. Because they're so special. There's such special circumstances. And to be fair, think about it. We probably don't want to test this ourselves because we don't want to. We will have to like expose humans, like have them sleep with the pigs, for example, have them get infected, and then we will have to take samples from them. I mean, that in itself is going to be very difficult and very dangerous. Now, in the laboratory, there are numbers as to how often, as to how easily this happens. And it looks like it is very easy for this exchange of genes to happen. But the laboratory is going to be very different than in the animals. All right. Any other questions? Now, being that we covered all of Unit 4 already, and you were tested on it and everything, you probably have heard somebody tell you. It's very likely that you have heard or read somewhere that someone said that they had the flu shot and the next day they had the flu. Let's talk about that. Is that even possible? Because usually what you read is that they tell you that they got the flu from the flu vaccine because they had it the day after they had the vaccine. And there's a movement of people that basically say to others, don't get the flu vaccine because you'll get the flu. Is that even possible? That's what I'm asking you. What do you mean by coincidence, Adam? Not because of the vaccine, correct? Because the vaccine cannot give you the flu. The vaccine cannot give you the flu because when we talked about it, we talked about how the shots include inactivated virus. So if the virus is inactive, then it cannot cause infections. Now, what if that person was incubating a flu strain when they got? So Daniel, the attenuated version is the nasal spray. And we usually give that one to children. So it is very unlikely that somebody got a shot with attenuated virus because we don't have shots with attenuated virus. Those don't get made. So it could be that they were incubating the flu already. There is an incubation period, and as we learn, there are no symptoms of disease when you're incubating at all. Now, could you give someone the flu from getting the flu vaccine? Even if you don't have any type of reaction, any type of fever, any type of malaise? No, because it is not an active virus. Now, could someone feel a little malaise from getting the flu shot like Liliana asked about earlier? The answer is sure, the same way if I don't know how many of you have had the chance of getting the second shot of the COVID vaccine. It's not fun. You could have some malaise, but it's not something that's going to last a week. This is something different. Sure, if you feel ill for a day, it's actually not a bad thing because that means that your immune system is actually kicking in. But that's not what we're talking about. We're talking about full-on flu that has been tested. A throat swab was tested, and it turned out to be positive. These persons may have waited a little too long to get their shot, and they were already exposed to some flu. Are there any questions before we move forward? All right, now something that we do care about quite a bit is the RSV, or human respiratory syncytial virus infections. Now, we don't see these ones in adults very often unless someone is immune compromised. Now, what we tend to see, and we know it has been reported, is that the virus could be present in an adult and they don't have any severe illness, but unfortunately, for those children that are not vaccinated, then the adult is shedding the virus and potentially exposing the children that way, which is why it's so important that we vaccinate young, very young, against RSV. Now, this virus is very interesting because in the fomites, it stays active for several hours. Most viruses really last 1 or 2 hours at the most outside of the body. But this one, we know already can be active 5 hours after. So that is a higher risk. That is another reason why washing hands when you're working with babies is very important. Now, at the beginning of the infection, they're gonna look and feel like they have a cold, a mild cold, but there's gonna be a lot of fluid production, and very quickly you will see that they will have inflammation of the bronchi, and they may even have fluid buildup, especially the babies. Toddlers may have a milder illness, but babies do not have such fate. So what happens usually is that they become anoxic because there's no good exchange of oxygen and carbon dioxide in the lung, and they end up having to be put on oxygen to make sure that they can get over without too much tissue damage. And the doctors may provide an antiviral in the form of ribavirin. Sometimes they're given just antibodies in an IV, which is what we have here, the palivizumab, which is just an antibody collection that we can provide the baby, because we know that if we don't do anything, the babies can potentially die from this. Now, we don't see nearly as many cases of RSV as we used to, and that is good news, and we believe that it is directly related to the vaccination campaigns. Now, if a baby has this, or the doctors suspect that a baby has this, then what we need to do is look for sometimes the virus in the bronchial fluid of the baby. And usually what is done is that it's mixed with antibodies to see if there's agglutination. If the baby has been sick for a while, then you will just look for antibodies against the virus in the blood of that baby. Are there any questions? Thank you. All right, so we're going to talk a little bit about hantavirus, which causes a condition that we refer to as HPS. Now, hantavirus is something that we see in some parts of the US where we have certain types of desert mice, like the one you see in the picture. The mice are not ill from this virus, they just happen to carry it. And when they urinate, they actually release the virus into the environment. That in itself is not a problem. It's just that the urine dries out. And when we walk through areas where these mice may have been at, you can potentially create aerosols of the dust that contains the virus. And if we inhale that, then we can develop this Hanta fever. At the beginning, we call it Hanta fever. You feel totally horrible, like you have the flu. But somebody that is a very healthy person, we're not talking about anyone that's immune compromised here, will not be able to get out of bed. They will look like they're going into shock. Basically, they have a spike in fever, their blood pressure drops severely. Some of them can just lapse into a coma. Usually, what is done is that, of course, they're going to be hospitalized and they are maintained, hooked up to all sorts of machines to try to keep their body going. The diagnosis for HPS is tricky. We tend to look at the big picture of what they're looking like in the hospital, where they may have been recently. Because it is an acute illness, it may take some time before you start seeing the antibodies against it. So serology can actually be done, but it has to be done multiple times to confirm it. And you could have a negative, and that doesn't mean much. And then later on, you realize, oh, well, yeah, it was positive. And what we do for these patients, unfortunately, is just kind of wait and see if they survive. Now, there are some experimental treatments against this, but that have been developed by some doctors in New Mexico, because we do see every summer cases of hantavirus in New Mexico, where they take the blood of the patient and they basically do dialysis in a way. And what they're doing is removing a lot of the clotted proteins that may lead to the patient to being in shock. Basically, you're not removing the virus, you're letting the body deal with it on its own, but we're just making it possible for the patients to survive the formation of all these clots due to the infection. There is no vaccine against this, so all we can do is be careful in areas where these mice can be present. I think it was 5 years ago, maybe less time, there was a hantavirus cluster that was due to some people that have been renting some of the cabins at Yosemite National Park, because there were some mice that have been crossing to the cabins and they have been peeing, and I guess the floorboards must have had the urine and it became aerosolized. Are there any questions? So, as far as, like, to give you maybe like an idea of a map. So when we talk about hantavirus, for the longest time, years ago, we used to say New Mexico, but we have had some possible, not confirmed hantavirus cases in some parts of Northwest, I should say West Texas more than Northwest Texas, where these mice can be found. But if you look at overall in the US, it's going to be in parts of the US, in the southern states where you have desert areas, where you can find this type of rodent. So we see it California, there have been a few cases, not that many in Nevada, New Mexico, some parts of Texas. It is usually lethal. I mean, it's something that can potentially kill you. So it's important that that supportive care is provided very, very soon. This is actually the picture of a desert mouse that I have here. Now, the good news, I know it sounds horrible. The good news is that this virus seems to be very sensitive to ultraviolet damage. So when it gets hit by sunlight, it becomes inactive. So that's one good thing. And also that it can only stay active in the urine for a certain period of time. So it tends to become inactivated very quickly. And it is not something that gets passed from person to person either. But yes, that viral PDOS is something that we need to think about. I mean, to be fair, DOS can contain other things like fungi. All right, I'm going to have to mute this because I can see a truck coming. I foresee noise. All right, they turn it off. My neighbors are getting some stuff delivered. They're getting a new deck. So sorry about that. And the dog immediately barked. All right, so let's just briefly look at other possibilities of infections that we could potentially get in the lungs, like something that we call metapneumovirus infections, and a few of the para-influenza viruses. Now, every flu season, we see some patients that have respiratory symptoms that seem to be maybe the beginning of influenza, but that doesn't quite develop like influenza. And it could be due to a metapneumovirus or one of the para-influenza viruses. Now, we usually just do a rapid test for influenza. And we don't have an easy, fast test for metapneumovirus or for para-influenza. And so we may actually just say, well, you have respiratory symptoms, but you don't have the flu. Once that flu test is negative, unless the doctor is really interested in learning why you have a certain respiratory condition, they may not really send samples for culture to the laboratory. Now, let's talk about where we normally find these metapneumoviruses. So there's usually two groups of them. We have one that we see mostly in birds, and then there's the other group that is the one that we care the most about, which is the human metapneumovirus. Now, the way it works out with these viruses is that they can be spread from person to person, but it turns out that in adults, the disease is not as severe as it is in infants. And we have a high mortality rate due to metapneumoviruses compared to adults. Now, because infants get RSV that we talked about earlier, sometimes the doctors make the assumption that it is RSV before they get the RSV test results as negative. But usually the children are treated the same way until we know otherwise. Eventually, though, you are going to be able to confirm that it was metapneumovirus because via serology, there may be antibodies that were made against the virus. Now, there have been some people that are adults that have had problems with metapneumovirus, but they were in very special categories. Maybe they already had severe asthma as adults, or maybe they had COPD. So when you have those pre-existing conditions and you get metapneumovirus, then that's when it's going to become more of a problem. Now, we tend to use something like ribavirin to try to help out those that have metapneumovirus, because we would have used metapneumo for the RSV ribavirin as well. So that's usually why we may end up actually throwing that at the patient. If you want rapid diagnosis of metapneumovirus that is not dependent on 2 weeks long culture, sometimes you can actually have a PCR test, but not all laboratories have it. So hopefully the doctors know where to send the samples to so they can get that test done. Now, parainfluenza is probably more prevalent than the metapneumovirus infections. And luckily for us, it tends to be relatively mild compared to RSV, metapneumo, influenza virus. Usually we diagnose this, unfortunately, by cell culture, so you end up having to wait. But it is a very good, reliable test. Only those that are very immune compromised or have lung illness get very ill from this every year. So whenever you say that your kid had a cold and they had a deep cough, more than likely it was a parainfluenza infection than a regular rhinovirus infection. It is very unlikely that you're going to even seek diagnosis because they get better on their own. Not much medication is needed. If the kids, if they're infants, they may again be given ribavirin, but for the most part, we just treat symptoms. Are there any questions? Okay. So we're going to move on to some of the fungal infections of the lower respiratory tract. Earlier, I kind of mentioned it a little bit, and I really was glad that the whole dried urine aerosols was mentioned because there are quite a few issues with aerosols that could potentially have fungi in it. There's some specific fungi that we are more concerned about than others. Now, to be fair, these are rare, but when we see them in patients that may be immune compromised, and it's very important that we can recognize that that is what they have. As far as blastomycosis, pneumocystis, and coccidiomycosis, those are the ones that we tend to see in those that are immune compromised that can be basically what ends up killing them. So we have to take these conditions pretty seriously. Now, to be fair, that first one, coccidiomycosis. So what we have here is a fungus that you find in soil. And in this map here, where you have the neon green color, those are areas in the Americas where we know soil aerosols can potentially have this fungus coccidioides. Now, we are potentially inhaling this fungus without getting sick. And it is only in those that are immune compromised that it becomes a problem. Usually they inhale the aerosols. Then in the lungs, you have basically just the growth of this fungus over and over and over and over again until the patients start developing a cough and they start developing maybe a little bit of chest pain. And what we see is that if it goes untreated, it can actually start going to different parts of the body, and you may even see skin lesions from it. And that is what you see in the picture here at the top. Worst case scenario is going to be that the fungus goes from the lungs to the brain. And usually at that point, there's not much that can be done for those that have developed the disease. Now, it is not something that develops super fast, and that's something to keep in mind. Some people have symptoms and then they don't, and they have symptoms and then they don't for several weeks, maybe even months. So it may happen that it's spread into the body without you realizing, but by the time you get to the doctor, it is very, very serious. Now, how do we diagnose this? You think, even though it is a rare disease, we want to make sure that we actually confirm that this is what's going on. So there is a type of test similar to the tuberculin test that we can do on the skin, where you will have a little, basically just blister that forms, that's reactive to, basically because you have antibodies, because you've been building them. It's very similar to the tuberculin test. And if you have a positive in that test, either you had the infection and hopefully you got better, or you are in the process of just having this disease spread to your body. Now, if it is early in the infection, that skin test is more than likely gonna come up negative. And so we have to just keep an eye on those patients. Maybe look at their chest x-rays, maybe do some CT scans, and maybe just try to see if samples can be taken to the laboratory to see if it will grow in culture. But it is going to take some time for that to happen. Now, once you have been diagnosed with this, then treatment is going to have to happen. There are a lot of different antifungals that can potentially be used. Depending on how advanced it is, it can be anything from 3 to 6 months worth of antifungal treatment. And you don't want to stop, because you have to make sure that you have wiped it out from all parts of the body. Are there any questions so far? can be this transmitted from person to person? The answer is that we don't have any evidence that it has in the past, so we say no. You have to inhale the fungal spores, and you have to be immune compromised. Now, what you're going to notice as we go on to the different fungal diseases is that there are many similarities among these, unfortunately. So basically, it's almost like I'm repeating myself over and over again here. So the next one that we're going to look at is going to be histoplasmosis. We have talked about histoplasma before. And in the case of histoplasmosis, you know that it is a fungus called Histoplasma capsulatum. We know that this one is also something that you can find in soil, but we have higher concentrations of this fungus, usually when you have accumulated bird droppings or maybe accumulated bat droppings. Now, where have we seen cases of histoplasmosis? Well, there are some areas in some central and eastern states where we have seen cases. We have along the Ohio and Mississippi River Valley seen some cases. It's not something that is exclusive to the United States. We know that it happens in some countries in Central America, in South America. It happens also in some parts of Africa and Asia. So it's something that can be just worldwide type of thing. More than likely, if you develop it, you breathe in the fungal spores, and most of us that have come in contact with this probably didn't even realize that had come in contact with, but some may develop a cough, maybe a fever, maybe you were a little tired for a while, and our immune system kicks in and we get protected and we get better. But for some other people that may have weakened immune systems, I'm not talking about immune compromise because that's obviously going to be a problem for them, then the infection can continue to get worse and worse and worse. And what they'll find is that they're short of breath, that they have body pain, that they have cramps throughout their body, and that it's something pretty serious. Now, the treatment for histoplasmosis, as you can probably imagine, is going to be an antifungal, like, for example, amphotericin B or an azole. If you do not treat it, like the one we just discussed previously, Coccidioides immitis, it can actually spread to the body, especially in those patients that are severely immune compromised. Now, for diagnostics, it's going to be very similar to what we just discussed. We're just going to try to get it to grow in culture. Sometimes you may be able to pick it up in a urine test, and the blood or urine test can be cultured for the patients. That is if it's become disseminated. All right. This one is also another one that probably most of us can have come in contact with, and we didn't experience any terrible symptoms from. It is Blastomyces dermatitis, which is the name of the fungus. Once you have developed the infection, it is something that we call blastomycosis. Now, this one tends to be one of those that we see in those that are immune compromised or older with a weakened immune system. It's also found in soil, but sometimes you can find it on maybe decomposing plant matter like leaves, trunks of trees. It is found in many different parts of North America, even in Canada. And what happens is that once more you breathe in the spores, and the symptoms are going to be mild fever, a cough, but then it can very easily get worse if you happen to be immune compromised. Now, this fungus can potentially spread through your body so that you have it in your skin, like lesions, you have it in your bones, you have it in your organs, you have it in your brain. So it is very important that if you have any type of symptom and you happen to be one of those people that are older or immune compromised, that you try to get diagnosed and treatment. Now, I had a question some years ago. Some student was like, well, what if I want to test soil around my house? Because now I think that there's fungus in my house or around my house, and I work in the garden all the time, and I may be inhaling spores of dirt that could have blastomyces or could have coccidioides. Or, I mean, you can imagine why this student was thinking that, because, hey, at this point, we're probably all thinking that. I was like, well, you know, like I said, most of us are inhaling these spores quite a bit, and we're not getting sick. It's only when you're immune compromised that it's a problem. And just because there is blastomyces in the soil, that's when just knowing that it's there is not going to be helpful at all, because there's nothing you can do to actually get rid of it in the soil, being that that is its natural environment. Are there any questions? All right, so we're going to talk about Pneumocystis pneumonia, which is something that for a really long time we did not know existed. That is the way it works out with a lot of these microbial infections. We started seeing cases of Pneumocystis pneumonia among patients that were in the late stages of AIDS. And we're still not confident of what types of environments actually carry this microbe. So the name of the organism is Pneumocystis jirovensii, and it looks like a yeast, but we suspect that it's probably one of those that has hyphae and also lives as yeast. We see that those that develop Pneumocystis jirovensii probably got it from the air, and we know that if you have multiple patients that are immune compromised, like let's say that you have 5, 6 patients in an area that have AIDS, it is completely possible for the infection to be passed on from person to person, and you can then have confirmed cases that were actually from human transmission. Now, it is a very tricky organism because when it was first found, and I'm going to make some circles here, here and here and here and here, all of those olive green structures are the actual fungus. And for a really long time, it was believed that it was a proteus, but it is not. It's a very funky shaped yeast. Now, there were some studies done in the late 80s, early 90s that basically were just collecting samples from a lot of different people, healthy people, lungs and respiratory systems, to see if we could find this fungus. And it turned out that there were many people out there, up to 20% of those that were healthy adults, that happened to have this fungus in their body, but they were not developing disease, that they actually had it in their upper respiratory system. And it was believed that many of us may actually develop mild illness and then clear it with our immune response. But for those that are immune compromised, then it is a different story. And then we actually have to do treatment of those patients. Now, because it's very difficult to grow this fungus in the laboratory, usually what it's done is that you have a biopsy done, and that's what this picture is from. You could actually have, sometimes if they're productive, you can collect some sputum and you can try to grow the microbe, but it's very slow grower. So what we do is that we extract DNA from the sputum, we do a PCR test, and this will find that pneumocystis DNA so that we can then confirm our suspicions. Now, usually what we have for treatment for patients is going to be a combination of trimethoprim and the sulfamethoxazole, which is, again, an azole, so it should work against fungi. Now, there are some issues with this drug where many patients, because it is sulfa-based, have an allergic reaction. So that can be tricky. And so we may have to try to use other antifungals that aren't as effective, but again, we don't want the patients to die from some allergic reaction. Are there any questions? Thank you. All right, so like I told you earlier, we're gonna just introduce the cardiovascular system diseases today. And remember that what we have been doing is that we try to look at what parts are concerned or are included, and then we look at them by type of microbe. So when we look at our cardiovascular system, we're talking about some structures like the heart and the blood vessels. And then we also take into consideration what's contained within them, which is going to be blood. And in that blood, remember, there's two parts to it. We have the clear portion, which is the lymph, and then we have our red blood cells. Now, there is crossover between our cardiovascular system and our lymphatic system. So some of the lymphatic tissues may be included as well. Now you remember that the heart is just a series of pumps that are able to move, create some pressure to move that blood throughout the different size blood vessels that we have. Now, one of the most diagnosed cardiovascular diseases out there is going to be septicemia, and there's different causes for that. And then we will look at other bacterial cardiovascular diseases like endocarditis, brucellosis, plague. This time we're going to be thinking bubonic, Lyme disease, and some tick-borne diseases in the form of ehrlichiosis and anaplasmosis. Now, when we look at this word, septicemia, immediately we think sepsis. So there's some sort of infection in the blood. Now, it could be that you have bacteria, literally bacteria, in your bloodstream. Or it could be that in your bloodstream, you're moving around a toxin that was produced by a bacteria somewhere else in the body. Now, we're going to start with this next time. Are there any questions before we pause for lab? All right. Well, I'll see you in 10 minutes then.